Today, our group will be presenting about rabies virus, starting with the content here. And this is our content. First, we'll be starting with the general characteristic. The rabies virus is classified in Rhabdoviridae family, consists of more than 100 single-stranded and negative sense non-segmented single-stranded RNA virus, and the common in the member of this family is a distinctive rod or bullet-shaped morphology. And the next one here is a scientific name. The realm is Riboviria, the kingdom is Orthonaviriae, and the phylum is Negernaviricota, and the class is Monoviricites, and the order is Mononegavirales, and the family is Rhabdoviridae. The human pathogen of the medical importance are found in the genera Lissavirus and Vesiculovirus. Only in the rabies virus, medically, the most significant member of the genus is Lissavirus.
For the rabies virus, is composed of an internal protein called or nucleocapsid and contain the nucleic acid. And the third one is have an outer envelope, and the last one a lipid containing bilayer covered with transmembrane glycoprotein spikes. And the next, this slide shows the sequence of single-strand RNA. And the next one.
Alright, so we're gonna talk about the pathogenesis. So you guys can look to this picture. Alright, so we're gonna start with a virus inoculate. And so we're gonna consider what virus gonna replicate in our muscle. And then the virion enter peripheral nervous system, and it's gonna have a passive ascent via sensory fiber. And then it's gonna replication in our dorsal ganglion. And then it's gonna rapid ascent in spinal cord, and it can infection of spinal cord, brainstem, cerebellum, and other brain structure. And so it's descending infection via nervous system to eye, salivary glands, skin, and other organs.
And as for the next one, we're going to talk about the rabies virus replication cycle. The rabies virus begins by attaching to the host cell receptor and entering via the endocytosis. Inside the endosomes, a drop in the pH triggers diffusion, releasing the viral RNA into the cytoplasm. The viral RNA is encoded and transcribed into the mRNA by the virus RNA-dependent RNA polymerase. The whole cell translates the mRNA into the viral proteins, while the new viral genomes are replicated using the persistent RNA template. The proteins and RNA assemble into the new virus, which branch off from the whole cell membrane, acquiring the lipid envelope. This is the process that rabies virus tries to replicate in the whole cells and can spread to the other cells. Next slide.
This part, I talk about the signs and symptoms. And next slide. This slide for the initial symptom. The first headache, fever, fatigue, chilling at sight of aperture. And the next. For the rabies specific symptom, hallucination, excessive salivation, light sensitivity, hydrophobia, insomnia, and aggression. And the next.
Okay, this part we will talk about laboratory diagnosis. Okay, for the first diagnosis for test is immunofluorescence staining. This test will use the sample types such as skin biopsy, brain biopsy, and we will use specific staining with C labels, rabies nucleocapsid, antigen-specific monoclonal antibody, and you can see the right-hand side picture that show the green one. This is the inclusions of rabies virus and antigen that are disclosed in cytoplasm of sensory nerves surrounding hair follicles. This is on the arrow ones appears apple green. Okay, for the next is the reverse transcription polymerase chain reactions, or RT-PCR. For the type of sample is saliva, skin biopsy, brain biopsy, and CSF. For the step, nucleoprotein gene will extract from the sample and will prepare smartomic. And next step is a reverse transcription step. We will heat to 40 to 50 degrees Celsius for 10 to 30 minutes, and we will add the primer. The primer will bind to the RNA template, and the enzyme that's called reverse transcriptase will synthesize cDNA by adding the NTPS, and after that the reaction will move to the PCR cycling that consists of denaturation, annealing, and elongation. This slide is shows the all three major steps for the RT-PCR. First is denaturation. All component mixture is heat to 94 degrees Celsius for 15 to 30 seconds. This step is the highest temperature. And double-strand cDNA is denatured to single strand, and this single strand will act as the template for the production of the new strand of DNA. Then for the next step, annealing. Annealing use the temperature between 54 to 60 degrees Celsius for 50 to 40 seconds, and the primer will bind to the target DNA sequence and initiate polymerization. For the last step, elongation or extensions. This step will occur at the 72 degrees Celsius, and the DNA task polymerase enzyme will sequentially add base to the 3' end of primer and extending the DNA sequence in the 5' to 3' directions. And DNA polymerase will add about the 1000 base pair per minute under optimal conditions. Okay, this picture is show about the result of gel electrophoresis, and you guys can see in the lane 1 was 100 base pair DNA rather, and in the lane 1 to lane 8 were all positive CSF sample. It means that there is a successful amplifications of the Rabies nucleoprotein gene, and in the lane 9 is a negative control, there is no bands, and lane 10 is positive control.
This slide shows the prevent and treatment. For the treatment, including the vaccinations of pets and avoid contact with stray animals, public education, immediate wound care, monitoring and reporting. Next. And this slide shows the treatment. The treatment is the wound care, rabies immunoglobulin or RIC, and supportive care. So in the rabies virus, there are no approved treatments for rabies. Once have symptoms, it will be exposed to rabies, and contact a healthcare provider as soon as possible.
Alright everybody, and this is our reference. And this is our group member. So thank you everybody. So if you have any questions, feel free to ask us. See you later. Thank you.